Hello everyone, Mr. Linder here. Let's talk about reabsorption in the nephron of the kidneys. So reabsorption is a mechanism to return molecules like water, sugars, vitamins, amino acids, and then electrolytes back into the cardiovascular system. So if we were to look at the general mechanism of the nephron, we could look at something like this. So blood comes in through the afferent arteriole, and then it enters the glomerulus, and then blood leaves through the efferent arteriole. And at the glomerulus and Bowman's capsule is where we have filtration. So we have what's called a GFR, a glomerular filtration rate. And filtration is driven by blood pressure. And so it forces solutes into Bowman's capsule and thus into the proximal convoluted tubule. The solutes that enter into the nephron can be both waste material, so it can be waste products, but it can also be things that we want to keep in the body. So we're going to have glucose, water, all kinds of electrolytes, vitamins, etc. So this filtrate that's being created through the filtration process has both waste material that we're trying to get rid of, but it also has materials that the body needs to keep. And so we have a mechanism of reabsorption back into the paratubular capillaries and into the vasa recta capillaries to take back the things that we don't want excreted from the body. And so that process is known as reabsorption. Turns out that reabsorption, about 99% of the filtrate that enters into the proximal convoluted tubule is actually reabsorbed back into the blood. Now, you still have reabsorption along the rest of the nephron, but most of the reabsorption is taking place in the proximal convoluted tubule. So oftentimes people ask, why not just filter out the 1% of waste material that needs to be eliminated? And really there's two reasons why the nephrons, why the kidneys work the way that they do. One of the reasons we don't just filter out the 1% is that waste material, foreign substances that are filtered from the blood, don't have mechanisms to be reabsorbed back into the cardiovascular system. So if there's no mechanisms for reabsorbing the vast majority of the waste that gets filtered, why not have a mechanism that has high filtration rates? And so we have a high filtration rate, so that we can remove waste material from the body quickly. And we can do that quickly because of that high filtration from the glomerulus into Bowman's capsule. And so the faster we move material into the proximal convoluted tubule, the faster it's being eliminated from the blood. And since the vast majority of foreign substances aren't going to have reabsorption capability, we can get rid of waste quickly. The second reason for not just filtering out the 1% of waste material is that it actually simplifies the regulation of water and electrolytes in the body. If we were to have excess, so if we have excess water, excess electrolytes, and you can fill in the blank here, excess vitamins, it's actually easier, again, because we have high filtration, it's actually easier to let that water and electrolytes and vitamins go into the nephron system and then only reabsorb what you need to keep and allow the excess to be excreted through the urinary system. So the two reasons have to do with the non-reabsorption of most foreign substances and the ease at which we can regulate the excess water, electrolytes, vitamins, etc., that end up into the nephron system by allowing for high filtration rates and then just reabsorbing what you need to keep. So what drives the reabsorption process? Well, essentially, sodium is either directly or indirectly involved in the transport of most substances. So really, it's sodium moving from the lumen into the extracellular fluid that becomes the primary driving force for reabsorption. So the primary driving force is figure out how to move sodium across the cuboidal epithelial cells of the nephron into the extracellular fluid, and then you can reabsorb everything else. So how do we move sodium from the lumen into the extracellular fluid? Well, there are transport mechanisms. And so in the cuboidal epithelial cells, there are epithelial sodium channels. So epithelial sodium channels, ENaC channels, that are allowing sodium to move from the lumen into the cuboidal epithelial cells. There's also going to be other types of transporters as well. There might be sodium hydrogen exchangers, etc. So we're moving sodium from the lumen into the epithelial cells. And then we use the sodium potassium pump to move sodium into the extracellular fluid against its concentration gradient. So we can pump sodium out and then bring potassium in utilizing ATP. So this is a primary active transporter. Because we run primary active transporters, that's going to allow for the secondary active transporters to function within the epithelial cells. So we start the movement of sodium, and then we can start driving secondary active transporters. So secondary active transport occurs because we have primary active transport. So here we have the primary active transporter moving the sodium into the extracellular fluid, and then that's going to drive the secondary active transporters. This happens to be just one example of a secondary active transporter. This is a sodium glucose transporter. So here we see sodium moving into the epithelial cell along with glucose. And so that's driving the reabsorption of glucose into the epithelial cells. But we could add other secondary transporters to give us an idea that we can move sodium into the epithelial cell along with amino acids. And of course, there's lots of different types of amino acids. So sodium with glycine, sodium with methionine, sodium with glutamate. We can move these amino acids into the epithelial cells through secondary active transport using sodium. We could also move ions. So sodium can move into the epithelial cells and then bring other ions with it, perhaps maybe bicarbonate ions, hydrogen ions, and so forth. So we see secondary active transport with sodium and ions. We see secondary active transport with sodium and lactic acid, sodium and citric acid. And so the list goes on and on. Once we have sodium moving across the epithelial cells, we then can use secondary active transporters to move sodium with glucose, sodium with amino acids, sodium with bicarbonate, sodium with citrate, etc., etc. So now we're starting to move other molecules into the extracellular fluid, and so we're driving the reabsorption of these other molecules. Once these molecules get into the epithelial cells, then you can use facilitated diffusion. So this is facilitated diffusion to move those molecules out of the epithelial cells and into the extracellular fluid. So again, this is just one example showing you the glucose that came into the epithelial cell. And then here we have a glucose facilitated diffusion protein that's allowing glucose to enter into the extracellular fluid. And so now we have reabsorption of glucose. But we could do this with amino acids. We could do this with other ions, other metabolites, and we are reabsorbing those into the extracellular fluid. Once we move sodium and we've moved positively charged particles into the extracellular fluid, now we can start driving the reabsorption of anions. So anions are going to be other negatively charged ions that can move across the epithelial cells and move into the extracellular fluid. So we are essentially allowing for opposite charges to be attracted. So perhaps a chlorine that's attracted to the positive charges in the extracellular fluid. So we're moving those negatively charged anions across the epithelial cells, and so we're getting reabsorption of those anions. Another thing to consider is the reabsorption of water. So as we move ions, so as ions enter into the extracellular fluid, as we move other solutes, so other molecules like glucose, like amino acids, okay, as we move those molecules and ions into the extracellular fluid, that's creating an osmotic gradient. And so now we see the reabsorption of water. Water is attracted to the high solute concentration. So water is going to move by osmosis from areas of low solutes to areas of higher solutes. As that water moves, it begins to dilute the extracellular fluid. So here we have extracellular fluid. We've had water now move across the epithelial cells. We begin to see a dilution effect. As more water gets into the extracellular fluid, that means leftover molecules and ions are becoming more concentrated. So we're seeing a concentration effect happening in the lumen of the nephron. And so as certain electrolytes and as certain molecules, even including some waste like urea, as those ions and molecules become more concentrated, those potassiums, calciums, they're able to move across the epithelial cells and thus be reabsorbed into the extracellular fluid. So even though we're trying to get rid of urea, there actually is some reabsorption of urea into the extracellular fluid as it diffuses across the epithelial cells. The last thing to consider for reabsorption, although it doesn't happen a lot, but there is some receptor-mediated endocytosis that takes place. And so receptor-mediated endocytosis is an active transport mechanism that's utilized by cells to bring larger molecules across the cells. So although we don't typically move proteins across the epithelial cells, because typically proteins are not being filtered at the glomerulus and Bowman's capsule, there are some small proteins and peptides that can enter into the nephron, and some of the epithelial cells are going to use receptor-mediated endocytosis to bring in those smaller proteins, digest those proteins into amino acids, and then move those amino acids into the extracellular fluid. So that's another mechanism for reabsorption of some small proteins and amino acids into the extracellular fluid. So the process of reabsorption, in summary, is to move sodium from the luminal space into the extracellular fluid, and in doing so, begin to move other molecules and ions. So we're going to move molecules, we're going to move other ions, we're going to move water, which is a molecule, but we'll designate that as its own. So we're going to move molecules and ions across the epithelial cells as we figure out how to move sodium, since it's the driving force for reabsorption in the nephron of the kidneys. So that is reabsorption. I hope that helps. Take care.